Thank you very much, Zila Raisna, for the introduction. I believe the same students are here that have heard me previously. For those online who meet me for the first time, let me introduce myself again. I'm Professor Naila Bulatova from the Department of Pharmacology and Clinical Pharmacology, Bashkir State Medical University. I'm adjunct here, and also I'm full-time professor at the University of Jordan, the School of Pharmacy, Department of Biopharmaceutics and Clinical Pharmacy. My pleasure is to give today my fourth lecture in the series of lectures about the COVID-19 pharmacological aspects of management. And in the today and the next lecture, I will be focusing on the investigational agents for the treatment of COVID-19. I will also briefly touch those agents that are in use. I will talk about their development, how did they reach the clinical practice. So some information will be a repetition of what you know from my previous lectures. Okay. Are you second or first year students? First year. Maybe something is difficult to understand, but it is a good opportunity, you know, to immerse yourself into the clinical reality and to see what is your future. Hopefully, we will be done with the pandemic very soon, but you know, this is the current reality that we have. Just to remind you that after the realizing that we have the coronavirus disease in Wuhan City, Hubei province, China, and its report to the WHO at the New Year Eve on December 31st, 2019. It was first declared as outbreak at the end of January 2020, but on March 11th, it was declared actually as a global pandemic when it started spread to the other countries. And this was first pandemic announced since the H1N1 pandemic of influenza that took place in 2009. Currently in the countries such as the United States and Europe, the only antiviral drug, as we discussed earlier, is remdesivir that is used in hospitalized settings. So this is the only antiviral drug approved fully in these countries. Other antiviral drugs are used, but they are within the frame of the clinical trials. Just to remind you that remdesivir in these countries is used for the treatment of hospitalized adults and children aged 12 years and older with a weight of at least 40 kilograms, and also the emergency use authorization, which is EUA, also allows to give the drug to pediatric patients weighing between 3.5 and 40 kilograms who are younger than 12 years. Or who are younger than 12 years who weigh at least 3.5 kilograms. Another treatment that was announced for the EUA, the Emergency Use Authorization, is the convalescent plasma. Last time I discussed this with you. This is the plasma that we obtained from the recovering people with COVID-19. And then, of course, after screening for the infections in this plasma, it is transfused to the people who are severely ill with COVID-19, providing the ready antibodies, hoping that these antibodies will bind the virus and eliminate the virus from the body. So this emergency use authorization for convalescent plasma was announced in these countries on August 23rd, 2020, almost a year ago. Also, we already know from the previous lectures that in the outpatients currently in the United States, there are several monoclonal antibodies, or we call them monoclonal directed therapies. Just to remind you that these monoclonal antibodies bind to the spike protein or S protein, which is responsible for the attachment of the coronavirus to the receptors, which are ACE2 receptors, the target of the virus, to be attached to the cell. So these monoclonal antibodies, include Sotrovimab, Casirivimab plus Imdivimab. You remember I told you one of them is used as solo, the first one. The other two are couples, such as Casirivimab plus Imdivimab, and the other couple is Bamlanivimab plus Etesivimab. These monoclonal antibodies, as we discussed, are used intravenously as a continuous infusion, but they are used in outpatients, which means that these people are only mild to moderately ill. Not to all outpatients they are used. They are used for whom? For those outpatients who test positive for the virus and are at high risk of severe COVID-19 or hospitalization. And I refer you to the previous lectures. I've discussed what are these risk factors for severe disease or hospitalization. For example, advanced age, obesity, the presence of certain diseases such as diabetes, etc., etc. So these are the structures, as you can appreciate from the image to the right. These are the structures of such monoclonal antibodies that attach to the SARS-CoV spike protein. I'm afraid to touch or to point to the microphone, to the screen. This is the spike protein, as you can see, and it is attached, the antibody is attached to this spike protein. All right. Also, some new immunotherapies. I would like to remind you that we have discussed in the previous lectures. Baricitinib, the JAK kinase inhibitor, was issued Emergency Use Authorization EUA on November 19th, 2020. It is not used alone, but always in combination with remdesivir. You imagine this is immunosuppressant. If you use it alone, we reduce the resistance against the virus. So we need to use it with antiviral drug, remdesivir, for the treatment of suspected or laboratory confirmed coronavirus disease in whom? In hospitalized patients. We remember that remdesivir is used for hospitalized patients, age 2 years and above, who require not every hospitalized patient, but severely ill hospitalized patients, those who require supplemental oxygen or invasive mechanical ventilation, or the more sophisticated technique, which is ECMO. As we discussed earlier, ECMO stands for the extracorporeal membrane oxygenation that treats respiratory and cardiac failure at once. So this is JAK inhibitor, baricitinib, or JAK stands for the Janus kinase, as you can see at the image to the right. This is the structure of baricitinib. And also this slide, the image to the right, shows how the baricitinib in red, you see the name in red, baricitinib, it controls the signaling inside the cell that is affected by the virus, and blocking this signaling inside the cell will prevent the release of the viral particles. So it has some antiviral properties, but also baricitinib, it modulates the release of the cytokines. You remember that damage of the cell triggers the release of inflammatory substances called cytokines. And you can see that these cytokines become elevated in the blood, including interleukin 1B, 1, 2. Let me enlarge it to show it. 6, 7, 8, 9, 10, 17. Also GCSF, which is granulocyte colony stimulating factor, GM-CSF, granulocyte monocyte colony stimulating factors, interferon alpha, interferon gamma, abbreviated as AIFN, TNF alpha, which stands for the tumor necrosis factor alpha, and some other cytokines. As we remember from the previous lectures, the release of these cytokines, inflammatory cytokines in the blood, are responsible for the cytokine storm. We have the systemic inflammation in the body, damage to the many tissues, not only to the lungs. We have the damage to the endothelium, which means because the endothelium is present in the blood vessels, and blood vessels are present in all organs. So basically we have the multi-organ failure in severe progressive disease. So this is the explanation. explanation of the why the baricitinib is used in progressive disease. I repeat, in hospitalized patients who require supplemental oxygen, because obviously in these patients we have cytokine storm and we have to block it using such immunomodulation. So these are drugs in use. I continue also about the next drug, which is now shown to be very, very successful. It is an interleukin 6 inhibitor, Tocilizumab, and we have studied already. You remember that interleukin 6 is now believed the main cytokine involved in the inflammatory storm or cytokine storm when the interleukin 6 binds to its receptor. Interleukin 6 is in green and its receptor is in blue. Such soluble interleukin 6 receptor that binds the interleukin now binds to the surface of the target cell, any cell, for example, the alveolar cell in the lung. And this causes certain signaling, by the way, including JAK kinase that we discussed in the previous slide. So this intracellular signaling, as we already know, is responsible for the release of the cytokines, the substances that cause troubles, inflammation, or cytokine storm. So by using Tocilizumab, as you can see, always MAB at the end of the word means that this is monoclonal antibody. So as you can see, the structure of monoclonal antibody is Y-shaped, right? Like English Y. In orange here on the right, Tocilizumab, the orange molecule, Y-shaped. Because it binds to the interleukin 6 receptor, it prevents the binding of interleukin 6 itself. So it blocks the actions of interleukin, no signaling, no release of cytokine, no cytokine storm. All right. So it was granted emergency use authorization in June 24, very recently, before a month, for the treatment of coronavirus disease 19 in hospitalized adults. So we are talking about the severely ill patients who have cytokine storm, also in pediatric patients aged 2 years or older, who are already receiving corticosteroids. Why do we need the addition of corticosteroids? There is certain explanation behind this, because clinical data show when Tocilizumab was used alone, it was not great. But when it was added to corticosteroids, then there was clear clinical benefit, improvement of the patient's outcome, like shortening of hospitalization, no need for ventilation, and prevention of death. That's why it is added to corticosteroids. And also in those patients who require supplemental oxygen, non-invasive or invasive mechanical ventilation or ECMO. Briefly saying those who are severely sick and those who experience the cytokine storm, obviously. Now, this was briefly about those drugs that have been already approved and are currently in use in many countries, including Russia. You know, the majority of these drugs, probably excluding the anti-SARS protein antibodies, they are in wide use in Russia as well. But now still we are in the urgent need for the treatments during this pandemic that such urgent need, it confounds the interpretation of resulting outcomes. And what I'm trying to say that normally the clinical trials should be conducted over a long period of time, very thorough examination of the results, clear statistical analysis. We need to get necessary number of patients. This is all not possible in many situations because we are in urgent need for these drugs. That's why we have the abbreviated clinical trials. Whatever positive we see, it is directly applied in the clinical practice. That's why the emergency use authorization, there is no full approval of the bodies such as FDA. So this all makes the interpretation of results of therapy difficult. And also, as an example of the number of compounds being evaluated, Gordon et al. In their articles identified more than now 332 high-confidence SARS-CoV human protein-protein interactions. Means that at the molecular level, we try to identify what happens when the virus attaches and then enters the cell and then is released by the infected cell. So we identified more than 300 targets where we can produce the drug intervention. And this is huge, and all of this is being investigated. And among these potential protein-protein interactions, there are 66 human proteins or host factors that we can target by 69 existing FDA-approved drugs in clinical trials and or preclinical compounds. What I'm trying to say here is that there is so-called drug repurposing. Repurposing allows us to repurpose drugs that are already in the clinical practice. For example, Favipiravir that we discussed at the previous lecture, so widely used in Russia and the Middle East countries. Favipiravir is a specific antiviral drug. It existed in Japan for the treatment of influenza resistant to other drugs, but it was not very popular because other drugs were safer. So it was standby for a long time. But when this pandemic appeared, the scientists tried Favipiravir for the SARS-CoV, and it was found to be successful in some studies, and that's why it is applied now as antiviral drug. What is this? Repurposing. The drug was used for some other purpose, but now it is repurposed for the treatment of COVID-19. And similarly, in the beginning of the pandemic, and I was talking about this, hydroxychloroquine, which is anti-malarial drug, or chloroquine, they were also repurposed. They were believed to be effective, but now it is proven that they're not effective. But these are all examples of repurposing. So we have 69 existing FDA-approved drugs that are repurposed, or we have some drugs in clinical trials yet, they are not used yet, or some preclinical compounds that are just synthesized or produced by biotechnology. And they are tried in vitro, as I said, then they will be tried in animals, and only after that they will reach the clinical stage. First the healthy volunteers, and then they will be tried in real patients suffering from COVID-19. I would like to mention here that WHO conducted a very huge clinical trial called Solidarity, mega trial, because it involved so many countries, so many medical centers. In this trial, the confirmed cases of COVID-19 were randomized. You know what is randomized. People with COVID-19, they were randomly assigned to different groups of treatment. And it was like lottery to ensure that there is no chance, or there is actually a chance to get into this group or the other. So one group was assigned to remdesivir treatment, the other for chloroquine or hydroxychloroquine. The third group was lopinavir, ritonavir. You remember this combination is used in the treatment of HIV virus or HIV infection, but now unfortunately the clinical trials show no effectiveness of this treatment for COVID-19. Or the fourth group was receiving lopinavir, ritonavir plus interferon beta 1A. In early July 2020, the treatment arms in hospitalized patients that included hydroxychloroquine, chloroquine, or lopinavir, ritonavir combination were discontinued. Why to continue this trial if we see that there is no effect? So these groups of people, they stopped receiving these drugs because there is little or no reduction in mortality when we compare with standard of care, oxygen, remdesivir, etc. And by the way, remdesivir was also shown not effective in the Solidarity trial, but now it is in the clinical use because subsequent trials, other further trials with better design have shown its effectiveness. But after that, disappointing results, it was not impressive. The remdesivir was not impressive. That was in mid-October. Interim results, it means results, sometimes we stop making the clinical trial and analyze. This is interim analysis, and then we decide to continue or not to continue. So there was no great result. Little or no effect on hospitalized patients in 4 groups of patients. And this was based on overall mortality. What is mortality? the prevalence of deaths was not changed with all these treatments, or initiation of ventilation. The same proportion of patients required ventilation with all these treatments compared to standard of treatment. And also they have noted the duration of hospital stay was not improved. It was the same. The 28-day mortality was 12% among hospitalized patients. Okay. That is regarding the existing treatments. Now I will start overviewing of the new things, new investigational drugs. And this slide I have displayed already on my first lecture. At my first lecture, I will repeat it again, that one point where we can interfere with the virus is to prevent its entry into the cell. You can see the virus, it's a spheric particle. It attaches to the specific ACE2 receptor, which its target on the alveoli or on other cells in the body. You can see the ACE receptor in the blue, its target. And also we have a certain serine proteases. One of them is TMP-RSS2. And we have other entry factors that help, as you can see to the right. The green TMP-RSS2 molecule helps the virus to be attached to its receptor. And after such binding, firm binding to the target cell, there is endocytosis. As you can see, endocytosis is engulfing of the virus surrounded by the host cell membrane. And then virus enters completely into the cell, and then it is uncoated, and etc., as we are going to see in the next slide. So this is potential target where we can block the attachment. And as we already said, we have the existing monoclonal antibodies that attach to the S protein. S protein is shown on this spheric virus particle as red projections. So we can block the entry, or we can block the serine proteases, those that I have shown in green. And also to the right, you can see another serine protease that can be also a target for the drugs called furin. Can you see the furin here? This is another serine protease that can be blocked by the promising drugs. So, but what exact proteases will participate in the viral entry will depend on the type of the cell. As I told you, the virus attaches not only to the alveoli of the lungs, it attaches also to the respiratory mucosa cell. You know that entry happens in the nose and oropharynx first. Then the virus may migrate to the alveoli of the lungs. And after that, the virus may go to the systemic circulation. As I told you, the endothelial cells located all over the body may be damaged to the virus through the same mechanism. So what particular entry mechanism will depend on the particular cell. But these are potential targets. I'm talking about promising strategies in the future that can be used to block the virus entry. Now, the next slide that we have already seen in the first lecture as well, following the uncoating of the virus, you can see that the endosome is formed, and then there is the release of the viral RNA. You can see the blue tape with the 5 and 3 end here attached to the host ribosome. So we have the uncoating, which means that the protein envelope of the virus is destroyed while the virus in the cell is in the cell. Then we have the, as I said, the release of the RNA of the virus into the cytoplasm of the host, the blue tape. Then we have the translation of the open reading frame 1A, or it is abbreviated as ORF1A, and also open reading frame 1AB to produce the polyproteins called PPs, PP1A and PP1AB. As you can see, they are shown in the orange as the orange tapes, PP1A and PP1AB. And these proteins are processed by the viral proteases. And these proteases are also encoded by the ORF1A. And this yields 16 non-structural proteins. Okay. So we have the formation of the RNA replicase transcriptase complex, RTC, below here. In this orange block, you can see RTC generation. It uses the rough endoplasmic reticulum, derived membranes, and the RTC drives the synthesis of the negative RNA. Negative RNA is shown in the red tape. Negative RNA is shown as the red tape. It is genomic in the right part of the slide. And the full length RNA copies, negative RNA copies of the genome provide templates for the full length positive RNA copies, which are shown below here. Subgenomic positive mRNA viral structural and accessory transcripts take place, and transcription further produces subset of subgenomic RNA, including those encoding all structural and accessory proteins. So after that, and by the way, here we can target several of these processes. For example, we can target the RTC, replication transcription complex. We can also block the viral proteases in addition to the host proteases, and any of these steps may be also targeted by the potential drugs. And after that, we said that we have the translation of the structural and non-structural proteins. Now we have the reassembly of the new viral particles. So these structural proteins and genomic RNA are assembled into the new spheric particles called viral nucleocapsid, and envelope is surrounding this nucleocapsid. Where does it take place? In the Golgi vesicle or compartment within the cell. And then these assembled particles, as you can see, whole particles are released by the process called exocytosis. Exocytosis, as you can imagine, is opposite to endocytosis. Endocytosis is swallowing. Exocytosis is the release of these new viral particles. Many particles are produced. So as you can see, the virus is very intelligent. It uses our cellular machinery, our cellular compartments to generate new particles and then to infect more and more cells. And also these steps may be blocked, as we are going to see. And this is the general view of the slide that we have split in 3 parts. Now we saw the yellow square by itself. We saw the middle part by itself, and we saw the right part by itself. Now all together is shown here. And as you can see, the below blocks are very essentially explaining the examples, how we can interfere with the virus in the future. The first yellow block, explaining the yellow block in the figure, we can block the entry, as I said. We can block the entry of the virus, right? How? By anti-spike antibody. And these are already in clinical practice in some countries, such as the United States. Or, okay, we don't have the anti-spike antibodies here in Russia, but we can use the convalescent serum and basically produce the same action. I already told you what is convalescent serum or plasma. Or prevention consists of vaccination. Have you received the vaccine or not yet? Excellent. So you are protected, hopefully. At least you are protected against the severe cases, which is the main thing. So this is because the vaccine provides the antibodies, but in contrast to the convalescent plasma that gives you ready antibodies, you form these antibodies yourself because you receive the antigen and now you produce the antibodies yourself, but basically you prevent the entry of the virus. Or as we said earlier, we can inhibit the serine proteases. For example, main serine protease, we know it, TMP-RSS2 can be blocked. These are drugs in the future. And we can also inhibit the endocytosis, theoretically. Endocytosis, we know this process already, the formation of the endosome. It may be blocked by the chloroquine and hydroxychloroquine. But as I told you, I would repeat again, the results were not great. And furthermore, the toxic doses were needed. Very high doses of these drugs are needed, much higher than we use in the treatment of malaria. And many people experienced cardiac toxicity. These drugs are toxic for the heart. And some cases where arrhythmias and even deaths from these arrhythmias occur. That's why chloroquine and hydroxychloroquine are not currently recommended. The second block, the orange block, blockade of replication, what happens in the replication transcription complex, or RCT, as we could see in the image. So we can either interfere with the RDRP, which is RNA-dependent RNA polymerase. And this is the major target for remdesivir. And by the way, similar drugs, similar action is produced by the Favipiravir, which is, as I explained, not used in the Western countries, but it is used in Russia, and many other countries. So this is the major mechanism, interference with the replication. How? I repeat, blocking the RDRP, RNA-dependent RNA polymerase, or also we call it replicase. Replicase is shown here as replicase polyproteins. All right. So this is done with the existing drugs, or we can block the viral protease. Can be blocked by, for example, ribavirin. Ribavirin, by the way, is existing antiviral drug. It long exists in the clinical practice. For example, it is used as inhalation for the treatment of severe respiratory syncytial virus disease, which may kill the neonates. Small babies, they may die from this virus, and they are given the inhalation of ribavirin. Ribavirin potentially may be used as viral protease inhibitor also against the COVID-19. And then, as we know, after the virus is released, and actually not only after the virus is released, when the virus damages the cell, it causes the release of the cytokines, as we know, called cytokine storm. And blocking the release of these cytokines may suppress the inflammation and serious consequences such as acute respiratory distress syndrome, requirement for the additional oxygen. All these things can be prevented by the immunomodulators, which is shown here in the next two squares. In the pink square, we have the boosters of innate immunity. And by the way, boosters of innate immune response. What is innate immunity? Probably you don't know yet. It is non-specific immunity. Some processes in the body that do not distinguish that this is particular virus and we need to deliver particular antibodies against this virus, or particular NK killer cells against this virus. No, this is non-specific immunity. Without recognition of specific pathogen, it directs the processes to kill it. So innate or non-specific, it attacks any foreign particle like the viruses, bacteria. Innate immunity includes the particularly promising is the interferon molecules. And I was talking about this a little bit. Please mute your microphone. Okay, so we can induce these interferon molecules. As you know, interferons are antiviral, and if we induce the interferon production in the body, like interferon 1 and interferon 3, this will boost the immune response, the innate immune response. And by the way, one of the theories that I like, I believe in regarding the COVID-19 is that those who get severe COVID-19 are those who have insufficient interferon in their body. They have certain probably genetic defects. They are unable to produce sufficient interferon, and that's why they progress to very severe disease. But if we use interferon, and as we discussed in the Russian guideline, the clinical recommendations are to use the intranasal interferon for the treatment of mild to moderate stages of the disease, and also for the post-exposure prophylaxis if you had close contact with the people with COVID-19, as we discussed. So using such interferon treatment or prophylaxis may be helpful. In other countries, interferon is still investigational. but in Russia, it is in clinical use. And enhancing of antiviral innate immunity. Also here, there is some repetition. I would emphasize importance of early treatment with interferon, or we can use the molecules that are interferon inducers. What is the meaning of inducer? It is not interferon itself, but it is small molecule that will enhance the release of our own interferon in the body. We have many such interferon inducers in Russia, but unfortunately, currently, they are not approved for this disease. They are approved for influenza, they are approved for other viral diseases. Maybe in the future, the clinical trials show effectiveness against the COVID-19 also of interferon inducers. Or new promising drugs are those that block interferon antagonists. Virus is very intelligent. It may induce interferon antagonists that will not allow the interferon to work in your body. But if you block these antagonists, we get the enhancement of interferon action. So blocking or blockade is somewhat very, very useful. Are you with me? Is it clear? Great. Okay. I have to look at the slide. In order to know when to make the break. Now I will talk about the remdesivir and its development, how it was developed. Sometimes the history is useful to know. Remdesivir, its trade name is Veklury by Gilead company, is indicated, I would repeat some information, for the treatment of only hospitalized adults and children aged 12 years and older with weight at least 40 kilograms. This is approved indication. But as we said, there is emergency use authorization. I will not repeat it. It is a broad spectrum antiviral. It is a nucleotide analog prodrug, as I explained earlier, and I will explain it again. Itself, remdesivir is not pharmacologically active. We call these drugs prodrugs. It is a precursor of a drug. But when introduced into the body, and I repeat, it is given by intravenous infusion, it converts to a substance, GS441524, which is an active molecule shown as an orange square here. Okay, and then it interferes with the RNA replication, as you can see here in the image. The GS441524 binds to the RDRP, which is RNA-dependent RNA polymerase enzyme. And this results in inhibition of the RNA replication, as you can see here at the lower panel of the slide. And to the right, you can see its binding pocket. If we enlarge, sorry, the molecule of the enzyme, RDRP, and this is the pocket where the drug is inserted, and when the drug is inserted into the enzyme, it blocks its action, so there is no RNA replication, as I said. No RNA replication, no multiplication of the virus. All right. Very expensive drug, as I mentioned. Approximately one vial is $520, and we need a minimum 5-day course, which is translated into more than $3,000 per 5-day treatment course. The history is very interesting. Why do we have the remdesivir so fast? I told you the drug development is usually very long, but fortunately remdesivir was already tried in Ebola infections, but against Ebola it showed very, very limited benefit. But then it was shown in the tissue cultures when we grow the cells in the culture, and then we add viruses to these tissues and see. It was tried against the SARS-CoV, which is severe acute respiratory syndrome. It happened in 2003, I told you previously, in the far eastern countries such as Vietnam, Philippines, maybe Hong Kong, yes, in these countries. By the way, it is very similar to SARS-CoV-2, which we currently have. Unfortunately, it had extremely high mortality, extremely high mortality. The chance to survive was very, very little. But why it was stopped very fast? Because the transmission was not as fast as with the current situation. The transmission could be limited quickly. Okay, so it was shown promising results against that virus in 2003. And then, if you remember, the Middle East Respiratory Syndrome, or MERS-CoV, happened in 2020 in Saudi Arabia and other countries. So it was shown promising results. That's why it existed in the market. And it was shown even in the animal models to be effective against SARS-CoV and MERS-CoV. And then it was quickly tried in the phase 3 clinical trials in many, many countries. And you remember I told you that Solidarity, which was the WHO trial, did not show good results, but further trials showed the very positive results. And the last was the real world analysis, which is, the remdesivir was used in real situation, not in the settings of the clinical trials, in real life. And these 3 retrospective, they looked in the past how the patients receiving remdesivir were doing after receiving remdesivir. This is called retrospective. They looked in the past. They were not designing the randomized trial. No, they just were experimenting in the real life. They looked retrospectively at the, and it was reported at the very recent World Microbe Forum. They showed that remdesivir treated hospitalized patients had significantly lower risk for mortality compared with matched controls. And this study was very impressive. Almost 100,000 patients were included. And convincingly, there was decreased mortality, which is the rate of deaths, I repeat, and significantly higher chance of being discharged from the hospital at the day 28. The patients recovered faster, and that's why they were discharged from the hospital to homes faster. And this is very good. That's why remdesivir now exists in most of the countries. And now I will talk about the drugs that are not in clinical use yet, but they show very promising results. One of them is from the Merck company, one of the leading pharmaceutical companies, and the drug is called Molnupiravir. Sorry, it's difficult to pronounce these names. Molnupiravir. Okay, and the previous laboratory names were used. It is an oral agent, and oral administration definitely much more convenient than the intravenous infusion. And it is like the remdesivir, it is also a prodrug, which means that it needs activation in the body and conversion into the nucleoside derivative, which is called N4-hydroxycytidine, or abbreviated as NHC. So as you can see, the M, the molecule of the drug, needs to be converted to the NHC, and then eventually it needs the phosphorylation into the triphosphate. So we have the NHCTP triphosphate. Okay. And then the further steps are similar to remdesivir, blockade of RNA-dependent RNA polymerase. So the net result is inhibition of the viral RNA replication, I repeat, very, very similar to remdesivir. So this is the same idea. So the drug is still in the phase 2A. Phase 2A is not the final step of the clinical trials. Do you want to have the brief idea what happens in the clinical development of the drugs? First of all, I repeat maybe this information. First of all, we study the drug in vitro, right? In biochemical reactions, with the enzymes, we add them to enzymes, certain enzymes, see whether it inhibits the enzyme of our interest or not. Sometimes we add it to the growth of the culture. We see whether the cells are growing or not, the virus or bacterial cell. Then in the animal models, for example, we can use the chimpanzee or some other primates and infect them with the human coronavirus and see how the drug prevents the development of the infection. These are all preclinical studies, right? Because we did not reach the human studies yet. If we get the very convincing results in these preclinical studies, then there are 3 phases of the clinical trials. The first phase We take very small group of volunteers, maybe several tens of healthy volunteers. These are healthy people. We explain that this is absolutely a new molecule and you might have the adverse effects. We just want to see the drug pharmacokinetics, how the drug reaches its level in your blood and see whether you experience adverse effects. Usually these healthy volunteers are paid good money because they participate in such research. And then using these several tens of healthy volunteers, we conclude how the drug behaves, how it is excreted by the kidney, by the liver, what toxicities it may experience, and so on. After good results of the phase 1 clinical trial, we go to the patients with this real disease. We care now about the COVID-19 patients, right? So we choose COVID-19 patients. And usually these are several hundreds of patients, and we see effectiveness of the drug and safety of the drug, which means we observe the adverse effects also. So we always care about two things: the efficacy, how the drug helps, and safety, how safe is the drug, whether it produces serious adverse effects or not. And after having good results from these several hundreds of people, we go to the phase 3, which is the final phase. And vaccines, by the way, were also experienced the same way, experimented in the same way. So in the phase 2A, it showed yeah, I might skip the details. Initially it was, please mute your microphone. Initially, the drug was tried in both hospitalized and outpatients, but inpatient study branch was halted, and in outpatients, it is still ongoing, and probably it will have the same place as the monoclonal antibodies. We will use it in outpatients who have at least one risk factor for poor outcomes or for severe disease, such as advanced age, as you know, obesity, diabetes. And fortunately, if fortunate result will be achieved, the patients will receive 800 milligram orally twice daily. Favipiravir, I have to stop here for a break. It's 11:48. After 5 minutes exactly, we will continue our talk. All right. If you have any questions, you can ask me, or you can prepare them and ask at the end. Okay, let us go on. The next drug is Favipiravir. It is in clinical use, as you know. Originally, it came from Japan as Avigan, as anti-influenza drug. It is approved currently in Russia, but not only in Russia, in many Middle East countries, for example, for the treatment of COVID-19. Maybe in India as well, I'm not sure. It selectively inhibits RNA polymerase, which is again necessary for the viral replication. As you can see here, the same idea as for the previous two drugs, as Remdesivir and the other drug from Merck. Favipiravir needs phosphorylation, and then when it is converted initially to monophosphate, then eventually to the triphosphate, Favipiravir RTP, it is triphosphate molecule. It blocks the RDRP. We know already that this is crucial step in the viral replication, RNA-dependent RNA polymerase enzyme. So the net result is the same as we use Favipiravir, and this prevents the viral replication, as you already know. The multi-center open-label randomized phase 2/3 clinical trial compared Favipiravir with standard care in Russia, and both two dosing regimens were tried. Both demonstrated good virological response. Viral clearance means that the body became free from the virus on day 5 was achieved in 62 or more percent, compared with only 30% of patients in the standard care group. So doubling of the percentage of people who were cleared from virus. And when they measured the viral clearance on day 10, then almost all people, 92.5% taking Favipiravir, were clear from virus, compared with only 80% in the standard care group. And this slide shows how similar are the actions of the nucleoside analogs. We call all these drugs nucleoside or nucleotide analogs. Actually, Remdesivir is nucleotide analog. It is a prodrug. Molnupiravir, as we said, it is also a prodrug, but it is a nucleoside analog. And Favipiravir is a nucleobase. But as you can see, all of them require phosphorylation, combination with phosphate in the body to become active. As you can see, we have 3 steps: monophosphate, which means one phosphorus group is added, a phosphate group, sorry, then diphosphate, and then triphosphate drug becomes triphosphate. This happens inside the cell, and then the triphosphate form of the drug incorporates into the viral RNA, and then it stops the viral replication. And we have some other drugs in the future, AT527 and Galidesivir, which have a very, very similar mechanism of action. So this is one of the promising approaches. Now also, as I told you, very much popular now is a repurposing of the existing drugs. For example, they took the antiparasite drug, Nitazoxonite. Nitazoxonite is already in use for the treatment of amoebiasis, for example. Amoebiasis is a parasitic infection, so it is a very well-known drug, broad spectrum, effective against many other parasites. And we have the extended release tablets, and they inhibit the replication of the broad range of respiratory viruses. As you can see in the right panel of the slide, it is effective against influenza A, HIV, rotavirus, HCV, which is hepatitis C, HB is hepatitis B virus, Ebola, Zika, sorry. You remember the Zika virus? Horrible virus. In Brazil, after the bites of mosquitoes, the usual individual is not affected. But if the lady is pregnant, then her baby has anencephalopathy, anencephaly, which means the absence or atrophy of the brain. So it is effective against Zika. And important for us, it was shown to be effective against MERS, Middle East respiratory, severe respiratory syndrome virus. And it acts here, in this case, against coronavirus, it acts against the viral N protein. You remember that in the first lecture, I was mentioning the N protein as one of the structural proteins of the virus. So it is now in 2 phase 3 trials for prevention of the disease, actually. Prevention, it means that we take the high risk population, for example, doctors, nurses who are working with COVID-19 patients, and we give them the drug to prevent them from being infected. Also, in the elderly residents, you remember that many elderly people who stay in the nursing homes, when the personnel of that nursing homes were coming and infecting them, almost all of these elderly people were dying. For example, in Sweden, remember that Sweden was using the model with no curfews, with no lockdowns, and they have lost so many people, including the people in the such homes. So hopefully this drug may be preventive. And in addition to the prevention studies, a third clinical trial for the early treatment of COVID-19 is planned. So this is a good example of repurposing. Next drug is Myclosamide. Myclosamide is very well known since I was a medical student as anti-helminthic drug. It is used in the treatment of helminthiasis, you know, helminths, those long parasites. Regarding the SARS-CoV virus, it is believed to disrupt the replication of the virus through the S phase kinase associated protein 2 inhibition. This protein is not shown here, but through this particular kinase inhibition, it prevents the autophagy and blocking endocytosis, meaning that the first steps of the entry of the virus, please see here. Remember, endocytosis and autophagy, which means that digestion of the virus takes place. Through this kinase inhibition, we prevent this step. And Myclosamide is now being in phase 2 clinical trial. All right. Other investigational drugs, we have 2 SARS-CoV-2 3CL protease inhibitors, both from Pfizer. And this is going to be very promising. The first drug is in phase 1B and the second drug is in phase 1 clinical trial. So they promise that by the end of this year, the drugs will be in clinical use. Where this particular protease is important, as you can see here, I've enlarged the image after the translation of the viral RNA. We have this protease that is essential for the proteolysis of the polyproteins, which are the precursors of the very important proteins that I was showing to them. So if we block this protease, we do not have the proteolysis and we have no formation of these essential proteins. So this is shown that we block both the 3CL protease and also non-structural proteins proteolysis. Hopefully we'll have good results in the clinical trials with this drug. Next, investigational drug. Now I will be mostly talking about the drugs that did not reach the clinical practice yet. They are in the clinical trials. We have the molecule called Enzovibep, which is a designed ankyrin repeat protein, DARP. This is a protein molecule. As you can imagine, this is a huge molecule. It contains, protein is genetically engineered like insulin, which means that, you know, the idea that the gene encoding for this protein is inserted into genome of the cells, for example, in the culture media growing Escherichia coli or some other bacteria. And then because this bacteria contains the gene encoding for this protein, it is generating this protein in large amounts. Then we purify this protein from the culture of the cells that generated this protein and use it as a drug. So this domain binds to the same epitope region within the SARS-CoV-2 spike glycoprotein RBD. RBD, you remember the receptor binding domain, the part of the spike protein that binds to the ACE2 receptor, but with 3 different antigen binding sequences. And this is now in the part of NIH, National Institute of Health trial called Active-3. And as I told you, in phase 3, we investigate both safety and efficacy in large scale in thousands of people. They are trying it in adults hospitalized with COVID-19, in addition to the existing standard of care, including remdesivir. So they combine it with the remdesivir. And to further clarify it, this is the spike protein in green. Okay, and we have the 3 domains of the DARPins, which are binding to this S protein and inhibit its action. Next drug, as you can see, we have tens of different approaches. Hopefully, at least several of them will be helpful. Rintatolimod. Rintatolimod is the toll-like receptor 3 agonist. Toll-like receptor 3, or TL3, is the receptor of the cells in the innate immunity. You remember, innate immunity is non-specific. It does not distinguish what particular causative organism is invading the body. So it is tested by the National Institute of Infectious Diseases in Japan at the University of Tokyo. And also it is tried for myalgic encephalomyelitis chronic fatigue syndrome, or ME/CFS, also called long hauler syndrome. It was announced in December 2020, and it is also a broad spectrum antiviral agent with immunologic properties. How does it act exactly? It acts on the toll-like receptor 3 here, shown in the green. And when we interfere with this receptor, we prevent the further signaling mechanisms, as you can see, downstream signaling, including the NFKB, which is nuclear factor protein. And then this protein induces the DNA transcription and the translation. And the next step is the inflammatory chemokines release. So by blocking these steps, the ultimate result is the blocking of the release of the inflammatory cytokines that we know are involved in the most life-threatening thing, which is cytokine storm. We block these events by blocking this early step. So this is a drug called rintatolimod, or Ampligen. Ampligen is the trade name. That's why it is shown here in the figure as Ampligen. I repeat, it is agonist. So it means that it does not block, but on the opposite, it stimulates the toll-like receptor 3 to block the certain signaling mechanisms that lead to inflammatory cytokine production. Next approach is to use the bemcetinib. IB or INIB at the end of the drug means it is inhibitor. INIB is inhibitor. It is the inhibitor used orally for the AXL kinase. And AXL receptor, as you can see in the image, it interferes with the entry and fusion of the virus with the endosomes that engulf the virus into the cell. And also, AXL receptor is important for the innate immunity, as you can see. And the bemcetinib may induce or block both steps of the viral entry, as I told you, and the release of the cytokines by the innate immune cells. And currently, the drug also is active in preclinical models against other viruses, including Ebola. You know, Ebola virus, dreadful, very dreadful, almost 100% mortality. It was an outbreak in Africa, in Central African countries. Fortunately, they have eliminated this virus. We do not have cases, but at any point of time, it may be active again. So it is effective against Ebola, against Zika that I have mentioned to you already. And recent data have expanded this to SARS-CoV-2. And currently it is in the phase 2 study. And it is done in the UK, in the United Kingdom, by the Accord initiative. Okay, next molecule is plitidepsin or aplidin. It is the member of the compounds known as diademins. They come from the plants. They are obtained from several herbs or plants, and in vitro studies from Spain report plitidepsin potentially targets EF1A molecule. And this molecule, as you can see here, sorry, it blocks the target EF1A. Let me enlarge it further, which is located in the ribosomes, so it prevents the multiplication of the virus by interfering with the mRNA transcription, by interfering with this part of the ribosome. And this is the key for the multiplication and spread of the virus. The other treatments include the VIR2703. As you can see, this drug is also existing as ALN-CoV. And in vitro data shows the drug targets small interfering RNA. As you know, in the cell, we have different types of RNA. We have mRNA, we have the small interfering RNA, abbreviated as siRNA. And the RNA interference, or RNAi, is the natural cellular process of gene silencing, and the siRNA molecules mediate, as you can see here in red squares, can mediate RNAi function by silencing the mRNA, which is essential for the viral replication that encodes for the proteins causing the disease. And their companies plan to advance development of the drug as inhalation. It will be used as inhalation. So this is a rare form. In most of the cases, we deal with the infusions and with the oral drugs. Next interesting example of the repurposing is the emitting hydrochloride. Emitting hydrochloride, when I was the medical student, and by the way, I was the medical student at this university, and these are the walls where I have studied, just like you. So when we were studying pharmacology, we were studying emetic. Emetic is an it is emetic drug, emetic drug, and that's why the name emetic. It is used to induce vomiting or emesis when it is necessary during the treatment of poisoning. When the poison was swallowed and you want to expel it from the stomach, you may induce vomiting. Of course, nowadays it is extremely rarely used, because now in the poisoning we use the gastric lavage, right? It is much safer to wash the stomach rather than giving the emetic drug and cause vomiting. But it is a very interesting drug, emetic, and it is obtained from the plants also, called the ipecac plant. So it is used as a syrup, sweet preparation. Okay, and also it is formulated as an injection to treat amoebiasis. Amoebiasis, as you know, is the protozoal infection. So then the clinical trials spread to viral hepatitis treatment and varicella zoster virus treatment, and other viruses, including Zika, Ebola. I will not mention all these viruses. So it seems that emetic is an antiviral drug, a broad spectrum antiviral drug. And it is also shown to be effective against different coronaviruses. And as you know, different coronaviruses in the past, only 2 of them caused serious outbreaks. But in the very, very past, when we were children, you know, coronaviruses usually are not harmful, and usually they induce a common cold, very mild illness in the childhood. So it was promising in the coronavirus's treatment, and now there is a phase 2-3 randomized blinded placebo-controlled multi-center trial in high-risk symptomatic adults with confirmed COVID-19 not requiring hospitalization. However, I'm critical. How you are going to use the emetic drug? It will induce emesis all the time. I don't know what's then. Maybe the doses are smaller than the emetic doses. I don't know what the scientists are thinking. Or you need to administer the very powerful antiemetic drug to prevent the vomiting from emitting, right? I don't know. Okay, next drug is the AT527 by another company. It is a purine nucleotide prodrug, shown here. AT527. It is effective not only against COVID-19, but also, as you can see, against hepatitis C, HCV virus. And because it is a nucleotide, it means that it is very, very similar in structure to the purines, those nucleotides that we have in our bodies. But because it is modified, it will interfere with the RNA polymerase. It inhibits the RNA polymerase enzyme. So it was shown both in vitro and in vivo for antiviral activity against several viruses, I repeat, including hepatitis C and coronavirus. Now it is in phase 2 study for patients hospitalized for moderate with moderate COVID-19. Another approach is to use the trabedersen. Trabedersen is the antisense oligonucleotide that inhibits transforming growth factor, TGF, beta-2 expression, and viral replication requires cell cycle arrest that is mediated by the viral induction of TGF beta, as you can see. So this inhibits TGF beta expression, and by this we cause the induction of the antioxidant defense. Antioxidant defense is very, very useful because usually during the infections or during the excessive activation of the immune system, the excessive oxidation damages the cell, and that's why we have the cytokine storm. But one of the approaches in the treatment of the inflammation is to produce the antioxidant effect by inducing several molecules such as cysteine, glutamate, gamma-glutamyl cysteine, and GSH, which is the glutathione. Glutathione, very well known to be an antioxidant component in the body. Next approach is to give the stannous protoporphyrin or SNPP. It is in the phase 2 trial for the treatment of COVID-19 in patients who are at high risk of deteriorating health because they are too old because of their age or because they have comorbidities. Comorbidities means that they have some other diseases such as chronic kidney disease or cardiovascular disease. And this slide shows how the drug acts. Protoporphyrin, here it blocks several steps like ferritin formation. Ferritin is the marker of inflammation, and hemoprotein causing tissue, eventually tissue injury. So we protect several tissues, particularly the heart and the kidney, by using this protoporphyrin drug. Next, I do not have the images for the next two approaches. The remdesivir is clear how it acts, but now the remdesivir is used also as inhalation because it's very difficult to give it by intravenous infusion. So they're trying in phase 1B to use the nebulized or inhaled remdesivir. It started in the last year in June, and they will use it in outpatients. Currently, as you know, it has place only in inpatient or hospitalized patient treatment. And the other drug is anthraquinonol. which is both antiviral and anti-inflammatory. So we both suppress the virus and we improve the immune system response. We suppress the inflammation. How? It reduces the nucleic acid replication similar to other nucleotides and nucleosides, as we could see. Viral protein synthesis is also inhibited and also prevent the tissue damage. As I said, excessive inflammation is limited. So it is now in phase 2 in patients with mild to moderate COVID-19 pneumonia. This slide came by mistake. I have to remove it because the discussion is here. For the first compound is apilimode dimesylate. It inhibits the lipid kinase apilimode. Please look at the figure. It acts on the lipid kinase enzyme called PIKFYFE, and this causes disruption of the lysosome that interferes with the entry and trafficking of the virus inside the cell, and it is in phase 2 still at the Yale University. Next approach is the use of Brekinar. It is orally available dihydroorotate dehydrogenase or DHODH inhibitor shown in vitro. Please look at the image below. When I'm trying to enlarge, it is shifted. Please yourselves look at the image in red, DHODH inhibitor in the image. It is shown to inhibit the viral replication and also broad spectrum of other RNA viruses. Why? Because the inhibition of this enzyme, DHODH, causes pyrimidine depletion. And as you know, pyrimidine are essential parts of the nucleic acids. And reduces the mitochondrial, you see this is the mitochondrial. It reduces mitochondrial electron transport needed for the viral replication. And it is now in phase 2 study in hospitalized patients, started in autumn last year. And brilacidine is the protein molecule actually, or protein mimetic, it is better to say. Protein mimetic, it causes the actions like protein itself. It has antiviral because it causes the membrane disruption in the virus, anti-inflammatory, which means that it has interference with the immune response. And the third property present in this drug is antibacterial property. And it is not a secret that for patients with COVID-19, we know that this is a viral infection, but when they are staying for the long time in the hospital, especially when they receive the mechanical ventilation, this is the source of bacterial infection as well. Hospital is always the source of the bacterial infection. And that's why these critically ill patients with COVID-19, they develop what? Bacterial superinfection. And then we start using antibiotics. Then when we start using antibiotics for a long time, there is spread of resistant bacteria and spread of fungal infection. And that's why the antibacterial property of brilacidine is very, very useful. And as anti-inflammatory or immunomodulating drug, it is inhibiting the interleukin 6, interleukin 1 beta, tumor necrosis factor alpha, and other pro-inflammatory cytokines. All of them we know to be the part of the cytokine storm. So it was very promising in vitro, and it will start being used in clinical trials. Vancomycin is in preclinical use. So we do not know the mechanism exactly. It is still in vitro at laboratory-based assays. Tempo, potential oral antiviral agent, and it limits SARS-CoV infection by impairing the activity of the viral enzyme RNA replicase. And phase 2-3 study is ongoing. And now I will very briefly in the remaining time talk about the immunomodulators and other investigational therapies. So far, we're talking about the antiviral treatments. Various approaches, as we already touched this topic previously, we can block the interleukin release. We can bind the interleukins. For example, we can bind the interleukin 6 molecule, preventing its binding to the interleukin 6 receptor, and furthermore preventing the steps of the activation of the immune cells and cytokine storm. We can block the Janus kinase, and we saw already this mechanism, or we can use the interferons. And one of the prominent immune modulator trials is started by the National Institute of Health, NIH, in October 2020. This is the phase 3 trial called Active Immune Modulators, Active IM, and they assess the safety of the 3 existing immune modulators. We have the Infliximab, Remicade. It is already used since long ago in the treatment of, for example, inflammatory bowel disease, such as ulcerative colitis. Abatacept, Orencia, and Cenicreviroc. The last one is the drug for hepatic fibrosis due to non-alcoholic steatohepatitis. So infliximab is the monoclonal antibody against TNF-alpha, tumor necrosis factor alpha. As I told you, this is a very important cytokine that may cause excess inflammation during advanced stages of COVID-19. And this is the place of the TNF-alpha in the activation of the macrophages and neutrophil recruitment. So these are all inflammatory cells. If we activate these cells by TNF-alpha, more and more cells will come to the site of inflammation. More and more cells will be involved. More and more substances of this type will be released. And this will cause the endothelitis. Endothelitis, as you can see, this is the damage of the endothelium of the blood vessels, which is the lining of the blood vessels, meaning that we have multi-organ failure. So blocking this response by blocking the TNF-alpha may be useful not only in the rheumatoid arthritis, psoriasis, inflammatory bowel disease, but also in the COVID-19. So it is currently in the trials. Abatacept is the T-cell co-stimulatory immunomodulator. We have the co-stimulatory signal here, as you can see, when the T-cell interacts with the APC. APC is the antigen-presenting cell. For example, we have the virus damaging the cell. Now the virus molecules are expressed on this damaged cell. As this damaged cell by the virus becomes the APC, antigen-presenting cell. And how does it activate the other cells of the immunity? By interfering with the T cell. As you can see on the second part of the diagram here, as a result of such stimulation or interaction between the two cells, we have co-stimulatory signal here, by the way. We have the MHC, which stands for the major histocompatibility complex, which presents the antigen, and the T cell receptor TSR from the T cell. So this is one interaction, but you can see also here the second interaction between the two other molecules, the CD80 or 86 and CD28 from the both cells. So after this interaction by two signals occurred, we have the resulting activation, proliferation, and production of inflammatory mediators, those that we are talking about, interleukins, TNF-alpha, interferons, etc., etc. So as a result of this, when we give the abatacept, we prevent the co-stimulatory signal, specifically where the orange is the abatacept molecule. It binds to the CD80 or 86 molecule, and it prevents its interaction with the CD28 molecule on the T cell. So prevent stimulation of the T cell, we prevent all the subsequent events. So this is a very promising treatment. So this is what I have explained already. And the third molecule, Cenicreviroc, a monomodulator that blocks the 2 chemokine receptors. 2 chemokine receptors. What are they? CCR2 and CCR5. And this slide shows that these two chemokine receptors are closely involved in the subsequent or in the consequences, of the COVID-19, very severe complications, which are ARDS. ARDS, it stands for the Acute Respiratory Distress Syndrome. When you see people on the TV or on the videos, they cannot breathe, they need the mechanical ventilation or other high oxygen requirements. This means that they develop this damage to the lungs, or ADRDS. How it is mediated by the cytokines, certain cytokines, or specifically in this case, chemokines, CCR2 and CCR5 receptors can be blocked by these drugs and prevent, as you can see here, it is written on this image, activation of these chemokines cause infiltration of neutrophils, meaning that more and more neutrophil cells, which are inflammatory, come from the distant parts of the body and attack their lungs, T cells and inflammatory macrophages. And the timely administration, as it is written here, timely administration of chemokine antagonists like this one, and anti-inflammatory agents such as dexamethasone, you already know about this, may prevent the chemokine upregulation and subsequent ARDS. So this drug, Cimikrivirok, is in clinical trial called One-Spy COVID-19. Interleukin inhibitors probably are the most widely used class of immunomodulators in COVID-19 treatment. And as you can imagine, they are not used deliberately in every single COVID-19 patient. No, when the patient reaches the hospitalized stage, not only this, when we see that he requires more oxygen support, including mechanical ventilation, we conclude, yes, this patient is in cytokine storm. Then we start giving these agents together with the remdesivir, which is antiviral, and together with dexamethasone, which is also very well known to suppress the cytokine storm. So such interleukin inhibitors of several classes, particularly interleukin 6, as we know, they may ameliorate severe damage to the lungs caused by cytokine release. I would like to mention again those cytokines that are particularly important during cytokine storm. Interleukin 6 probably is the most important. Also interleukin 1, that's why in Russia, interleukin 1 receptor antagonist is in use. Also interleukin 12 and 18, and tumor necrosis factor alpha. We were talking just now about the infliximab that is in clinical use since long ago as an anti-TNF alpha drug. So as I just explained, the release of these cytokines causes inflammation in the lungs, pulmonary inflammatory response. And this causes the decrease in alveolar capillary gas exchange, making oxygenation difficult in patients with severe illness. Here, as mistake should be, may result in decreased alveolar capillary gas exchange. We do not have the exchange of the gases. So no oxygen in the body, severe illness. I will probably skip this slide for the sake of time, and I would rather focus on the Tocilizumab. We have mentioned Tocilizumab last time. It is in all clinical guidelines, in the Western clinical guidelines, in the Russian clinical guidelines, because very recently it was shown to be a very powerful interleukin 6 inhibitor in COVID-19. By the way, Tocilizumab, why fortunately we have it? Why do we have it already? Because fortunately, it is used for many years in the treatment of rheumatoid arthritis. You know, rheumatoid arthritis is one of the autoimmune diseases. And it was shown that the interleukin 6 is also important for rheumatoid arthritis. And that's why we have this drug. And then it was, you know, in the beginning of the pandemic, we were afraid that people with autoimmune diseases such as rheumatoid arthritis, they receive immunosuppressant drugs such as interleukin 6 inhibitor, Tocilizumab, they might have poor outcome. They might die more frequently, or they may have more serious disease. No, the opposite was shown, that they had less severe course of the disease. Why? Because they received interleukin 6 inhibitors. And from here, from this observation came the clinical investigation. And now this drug has very firm place in the treatment of COVID-19. So as we already know, the COVID infection causes dose-dependent production of interleukin 6 from the bronchial epithelial cells and from other cells of the body, including lymphocytes, monocytes, and fibroblasts. And Tocilizumab was issued as emergency use authorization on June 24th. See, very, very recently, just 1 month ago, it received not approval, but EUA. I told you several times what is EUA because this emergency situation. For whom? For whom? What is the place currently? I repeat, for hospitalized adults and pediatric patients aged 2 years and older with COVID-19 who are already on systemic corticosteroids. Not every single patient, no. When the dexamethasone is already used and this patient already on supplemental oxygen or non-invasive or invasive mechanical ventilation or extracorporeal membrane oxygenation. So it means that these patients are the most critically ill, then we start using this drug, not to every single patient, of course. This is just to show again, maybe I was showing that more than once, how the interleukin 6 is involved. So this is the virus, SARS-CoV-2. The spheric particle, it activates the macrophages, dendritic cells, and monocytes. This is innate immunity cells. They release interleukin 6. Interleukin 6 binds to the receptor shown on the surface of the any cell, maybe alveolar cell. Binding of the interleukin to receptor induces intracellular signals, including JAK-STAT pathway that we already know and other pathways. And this releases further cytokines, causing cytokine storm. So how the Tocilizumab acts is shown here. It blocks the binding of interleukin 6 to its receptor and prevents, therefore, the cytokine storm. What particular receptor, what particular mechanism, it is under question yet. Our time is finishing, and I will just finish about the Tocilizumab. Now it is recommended in addition to standard of care, I repeat, in addition to steroids among hospitalized adults who have elevated markers of systemic inflammation, for example, elevated C-reactive protein, elevated ferritin, and other markers of inflammation. This is according to the Infectious Disease Society of America, which is IDSA guideline. Another American guideline, the NIH, which is the National Institute of Health guideline, recommends its use also in combination with dexamethasone as a single IV dose. It is a single dose per course. With dexamethasone in recently hospitalized patients who are exhibiting rapid respiratory decompensation. In other words, as I said, those who require supplemental oxygen, including mechanical ventilation and ECMO. And this recommendation is based on the positive. We have very little evidence coming from the clinical trials to show certainty of mortality reduction. We are not 100% certain about this drug. That's why, but because it might be crucial life-saving, such a recommendation exists. So I will just mention that results of these trials are controversial. One group of trials were showing that there was great impact on mortality. It prevented the deaths and it improved the hospital discharge. But in the other trials, Covacta trial, there was no improvement. And that's why the current recommendation is very, very cautious. It is to use it, I repeat, only with dexamethasone and only in those who require supplemental oxygen. And there is another interleukin 6 inhibitor, Sarilumab, but we have only preliminary results, and that's why it is not in clinical use yet. I would stop here because the time is over. And please remind me that next time we'll talk about the interleukin 1 inhibitors, and we'll continue in the subsequent lecture our talk about the investigational treatments of COVID-19. I hope it was quite clear, although it is a very complex topic, and I realize that for the first 2-year student, it is very challenging. But I wanted just to introduce you that we have many, many promising results, and hopefully in the future we'll have the solution to this disease. Any questions, please? So far clear? Maybe online questions, please. We can handle a couple of questions. If the patient had the COVID-19 already in the recent past or currently in the recent past. As I told you in the first lecture or in my second lecture about the vaccines, the countries differ in their recommendation. In Russia, current recommendation, I believe, is to give after 6 months. In other countries, like in the US, they say after your infection is over and you stopped being infectious to the others, let us say after 14 days, just go and do the vaccine. Very good point. And actually, we might have the answer to this question after 1 year or after 10 years. I don't know for sure. But, you know, there are several opinions. First of all, not any patient develops antibody response. We might see negative or very low titer of antibody. Second, because of new mutants, you know, variants of concern or VOC, such as Delta variant, which is now predominant in most of the countries. Because of these new variants, the level of antibodies that are produced, let us say, hypothetically, you were sick with the, you know, ordinary mutant, the previous variant, traditional variant, okay? And you have antibodies, very low titer, relatively low titer, not like after the vaccine. You have low titer, and this titer of your antibodies might be not sufficient to kill or inactivate the Delta variant. This is the rationale for going for the vaccine. For example, after Pfizer, my husband had Pfizer in Jordan, his antibody level is 700 something. Me, I had Sinovac, you know, in Jordan, it's like lottery, not the vaccine that you want, but the vaccine that is available today. We register on the platform and then you are called on a certain day. Today is Pfizer, tomorrow is AstraZeneca. My day was Sinovac, which is Chinese vaccine, inactivated virus, just traditional platform. So I checked my antibody level 1 month after vaccination, it was 70. Now I've checked it is more than 6 months after vaccination, it is still 70. Thank God. But other people immediately after vaccination or immediately after the disease, they do not have any level of antibodies or they have very low level of antibodies. So when you compare the level of antibodies after disease in general, level of antibodies after disease is much lower than the level of antibodies after the vaccination. And that's why they recommend still after the disease to go and to do the vaccination to increase your titer of antibodies. But we don't know. Maybe it is not necessary to have this high titer of antibodies. Maybe you have the memory cells, which is cellular immunity that you cannot measure with the current methods. We can only measure the antibodies, right? Now, to measure the T cell immunity or memory T cells, you need to go to the very sophisticated lab, and this is very, very expensive, which is done only in the research labs currently, not in the clinical labs. So this is the rationale. I hope that I've answered your question. Okay, you're welcome. Any other questions? Yes, because the other trials, as I was covering, they did not reach the approval. You have to finish the stage 3 or phase 3 clinical. Yes, this is a good point. As I told you, okay, trials, no, the trials were concluded, they were completed, but there were controversial results. Solidarity trial was not showing any positive results while other trials showed. But the reality in real situations that they have shown positive results with remdesivir. That's why it is used. Okay. When we are talking about EUA, Emergency Use Authorization, this means that, okay, we are desperate to give this drug, but this is not approval. But we said that remdesivir is approved. For example, in the US, it is FDA approved, which means that it has completed all phases of clinical trial. And we have this approval in adults, hospitalized adults and children above the age of 12, as we said. Approval means complete trials conducted. Okay. Other questions? Online questions? I think we are exceeding time. Okay, thank you very much and see you after tomorrow. Bye-bye.
Thank you. Goodbye.
Thank you.
Thank you so much.
Мы завершили. You are welcome.
Good day, dear students. I will welcome you to our educational school for pharmacy students. Today is our last lesson of this summer educational school. My name is Artur Khalyafovich. I'm an associate professor of the Department of Management and Economy of Pharmacy. We studied with our students of the first year the course of history of pharmacy, and we, as you remember, we studied the course of the basis of legal basis for pharmacists with students of the second year. So today we will speak about circulation of medicines in pharmaceutical distributors and pharmacies in the Russian Federation. And today we will watch with you two videos. Firstly, let's watch the first video. The first video will show the work of one of the largest pharmaceutical distributor in the Russian Federation, Katren company. A pharmaceutical distributor is a large wholesale company that sells medicines and other medical goods for hospital pharmacies, for community pharmacies, and hospitals, of course. After this video, we will watch the second video about the work of pharmacy, and after watching these videos, we will consider a lecture about the international guidelines about good pharmaceutical practice. So, firstly, let's show one short video material about work of the largest pharmaceutical distributor in the Russian Federation. The video file will be in Russian, but don't worry, please. I think the information in this video will be available for you, and we will comment this video after watching it. So let's watch this short video about the work of pharmaceutical distributor of specialized wholesale company that sells medicines for to pharmacies and hospitals in the Russian Federation. So let's watch this video.
Уже 2 десятилетия подряд компания Katren работает в интересах своей страны. Сегодня, благодаря нашей деятельности, миллионы россиян получают необходимые лекарства там, где им нужно, быстро и по доступной цене. 15 лет особое место в созвездии Katren занимает его уфимский филиал, официальный представитель крупнейшего национального фармацевтического дистрибьютора, известного далеко за пределами нашей страны.
Компания Katren находится в постоянном развитии. Растут обороты, объемы, планы и задачи. Вместе с компанией растет и наш филиал в городе Уфа. Сегодня мы располагаем высококвалифицированными кадрами и всеми необходимыми ресурсами. У нас трудятся те, кто заинтересован в успехе общего дела и компании Katren.
Время основания филиала – 1999 год. Профиль деятельности – оптовые поставки лекарственных средств от лучших производителей. Персонал – 132 человека. Территория охвата – республики Башкортостан и Удмуртия. Сейчас уфимский филиал компании «Катрен» вбирает в свою орбиту до 30% регионального профильного рынка. И это не предел. Гарантия успеха нашей работы – растущее доверие со стороны клиентов, их уверенность в качестве и уровне предлагаемых нами услуг. Кроме того, мы единственная крупная компания-дистрибьютор, располагающая собственной развитой складской инфраструктурой на территории региона.
Компания «Катрен» не случайно занимает лидирующие позиции на региональном рынке. Наша безупречная репутация и логистика, постоянно растущий ассортимент, гибкая система расчетов и реализация совместных проектов позволяет нашим партнерам грамотно строить свой бизнес и служит лучшим залогом привлечения все новых и новых клиентов.
Партнеры филиала – более 1900 аптек региона, лечебно-профилактические учреждения и медицинские центры. Ежегодный прирост по продажам не менее 30%. Площадь склада – 7500 квадратных метров. Склад уфимского филиала компании «Катрен» – самая многочисленная служба филиала. Это грамотные специалисты, это автоматика, это контроль, это собственная программа логистики. И, конечно, это новые, просторные и современные складские помещения, а также предмет особой гордости компании, введенный в 2015 году современный автоматизированный сборочный конвейер.
Переезд склада на новой площади – это явление актуальное и назревшее. Мы говорим о новых условиях работы, о мощном развитии компании в рамках родного региона, о прогрессивных технологиях складирования, оптимизации процессов контроля, сборки и доставки заявок до клиента. Это еще один шаг вперед, это еще один шаг в будущее.
В 2002 году ассортимент лекарственных препаратов на уфимском складе компании насчитывал 2500 позиций. В 2004 – 6000 позиций. К 2015 году данный показатель удвоен и составляет более 12 тысяч наименований. Работы автоматизированного сборочного конвейера от всемирно известной фирмы КНАП говорит сам за себя. Набирает обороты скорость обслуживания клиентов. Растет пропускная способность склада за смену. Больше, лучше, качественнее, быстрее. Можно не сомневаться, каждый из обрабатываемых здесь заказов будет доставлен в точку назначения строго в оговоренный срок. Выгодно и просто.
Пуск новой производственной линии упростил технологию сборки заказов. Прежде всего, серьезно выросла производительность труда. Она выросла в 3 раза. Месяц назад мы собирали 2000 позиций в час. Сейчас собираем 6000 позиций в час. И это еще не предел. А это значит, что мы принесем еще больше пользы нашим партнерам, доставляя медикаменты точно в срок.
В интересах клиентов филиал реализует программы «Катрен Финанс», «Дельта-10» и «Аптека.ру». Это беззалоговые займы на развитие бизнеса. Это рост товарооборота и рентабельности. Это привлечение новых покупателей и улучшение качества обслуживания. Пока мы путешествуем по уфимскому филиалу компании «Катрен», сюда с разных концов Башкирии и Удмуртии поступают десятки и сотни новых заказов. Это реальный факт. Мы делаем все, чтобы нашим партнерам было удобно с нами работать. Время продолжает свой бег, а наш рассказ подходит к концу. За кадром осталось немало важных событий. От благотворительной помощи детским домам до многолетнего лидерства среди региональных дистрибьюторов. Все охватить нельзя. Но, как и 15 лет назад, мы продолжаем динамично развиваться. Стремимся к лучшему, опираясь на достигнутое. Уфимский филиал. Компания «Катрен».
Нам 15 лет. Важнейший фактор нашего успеха – уважение наших сотрудников, партнеров и клиентов. Я благодарна и признательна за бесценную поддержку и доверие. Хочу пожелать всем успехов в бизнесе, процветания и благополучия. Отдельное спасибо тем, кто не дает нам расслабиться и стимулирует нас к лидерству и непрерывному совершенству. Все только начинается.
Okay, very good. I will repeat that you watched now the work of Hello. You watched the work of one of the largest pharmaceutical distributor in the Russian Federation, the Katren company. And I want to say to you that the filial of this company is located in our city, in Ufa. And I think that we, with you, with our pharmacy students, will go and we will visit this company in Ufa in our senior courses. Yes, so because it's very interesting. For example, in this video, you watched the green plastic containers, yes, for medicines. The employees of this pharmaceutical distributor package medicines into these containers, and then these containers with medicines are delivered to pharmacies, to our pharmacies and our hospitals in our city. So it's very convenient for our pharmacies, for healthcare workers, and I think that we will consider these interesting processes in more detail in our senior courses. Okay, now let's start to consider of our. Okay, yes. Здравствуйте, hello, yes, please. Okay. Let's start to consider of our lecture. Today, I want to say with you about the guidelines of International Pharmaceutical Federation and World Health Organization about good pharmacy practice, GPP, good pharmacy practice. Nowadays, we can say that medicines are an essential and critical part of healthcare services in all cultures and societies. When accessed, medicines are often an essential component of many disease prevention programs and virtually all disease treatment plans. And of course, we should consider the role of pharmacists in the healthcare and especially in good pharmacy practice around the world. We can say that pharmacists are healthcare professionals, of course, whose professional responsibilities and accountabilities include seeking to ensure that people derive maximum therapeutic benefit from their treatment with medicines. And so this requires them to keep abreast of developments in pharmacy practice and the pharmaceutical sciences. Also, we should have the special professional standards and requirements in every country. And of course, the legal regulation, yes, the laws governing pharmacy and medicines and advances in knowledge and technology relating to use of medicines. And of course, we can say that pharmacists are specifically educated, of course, and trained health professionals who are charged by the national authorities with the management of the distribution of medicines to consumers, including the work of the wholesale companies, yes, pharmaceutical distributors, yes, you watched the video about the work of pharmaceutical distributor, and Of course, pharmacists can engage in appropriate efforts to assure the safe and efficacious use of these medicines, of course. And as healthcare professionals, pharmacists play an important role in improving access to healthcare and in closing the gap between the potential benefit of medicines and the actual value realized and should be part of any comprehensive health system. Also, in addition, the increasingly complex and diverse nature of pharmacists' roles in the healthcare system and public health demands a continuous maintenance of the competence of pharmacists as healthcare professionals, of course, who have up-to-date skills and expertise. Of course, in Russian Federation and in other countries too, of course, pharmacists and other healthcare workers should update their skills every year, yes, and should pass the special procedures, maybe exams, every 5 years. And so let's speak about the mission of pharmacy practice. The mission of pharmacy practice is to contribute to health improvement and to help patients with health problems to make the best use of their medicines. And according to the international guidelines of International Pharmaceutical Federation, FIP, and the World Health Organization, we can say about 6 components to this mission. Firstly, being readily available to patients with or without appointment. Secondly, identifying and managing triaging health. Or triaging health-related problems. Thirdly, about health promotion. Later, about assuring effectiveness of medicines, including preventing harm from medicines, and of course, making responsible use of limited healthcare resources, because you understand that in every country, healthcare resources is limited, unfortunately, yes, but we as future healthcare future healthcare workers should work in these conditions, and you can choose the optimal effectiveness medicines for treatment our patients. Especially we should consider the situation with community pharmacies. Yes, in community pharmacies are working for selling medicines to patients. Yes, I think you saw these pharmacies in Ufa and in other cities too. So we can say that in the community setting, pharmacists should be acknowledged as healthcare professionals whom patients can consult for health-related problems. Because healthcare products and services are available from the pharmacist, some problems can be managed at this point of care. Yes, we can say that in some situations, patients go firstly to pharmacy for consulting with pharmacists. Yes, before going to doctor, to physician. Yes, so we as healthcare professionals should be prepared for this situation, and we can consult. We can consult our patients as pharmacists, as healthcare workers for improving the situation with health of our patients. Yes. Also, we can say that problems that require additional diagnostic skill or treatments not available from a pharmacist can be referred to an appropriate healthcare professional or site of care, such as a hospital, firstly, of course. But we can say that in any case, it should be done in good collaboration between the healthcare providers, especially between doctors, physicians, nurses, and of course pharmacists. And to improve the use of medicines, pharmacists have responsibilities for many aspects of the process of medicines use, each of which is important to achieve good outcomes for treatment of our patients. And also we can say that this begins with assuring the integrity of the medicine supply chain, including detecting spurious, falsified medicines, ensuring proper storage of medicines and quality preparation of medicines when needed. Of course, we in our department and in other specialized department of pharmacy faculty of pharmacy school of Bashkir State Medical University, we study with our students the many aspects of circulation medicines in pharmacy too. For example, in our department, we will study the legislation and international regulation for storing medicines, because you as future pharmacists should understand how to choose optimal temperature for storing medicines in pharmacy. And of course, we as pharmacists can consult our patients about rules of storing medicines in patients' homes. Yes, of course. So yes, these aspects will be studied in senior courses. Yes, in our faculty. On our faculty. Also, we can say that the optimal, the good pharmacy practice also includes assuring the proper prescribing of medicines by doctors, of course, by physicians, so that those regimens and dosage forms are appropriate. Instructions for use are clear. Medicine and medicine-food interactions are prevented, known and predictable. Adverse medicine reactions, including allergies and other contraindications, are avoided. Unnecessary treatments and minimized, and the cost of medicines is considered. Yes, if it's available in such situation. Yes. Also, of course, for example, you will study the medicine-food interactions in the course of pharmacology, and also we will repeat the main aspects of these interactions in our department in the course of pharmaceutical consulting of patients, of course. And also in senior courses, in our department, we will study the organization of working in pharmacies with potential contraindications in using of medicines. Yes, for example, we will study the monitoring of safety and effectiveness of medicines. In other words, excuse me. In other words, we will study the basics of pharmacovigilance. Yes, and because our work as pharmacists includes the working and if it's available, the treatment of contraindications during using of medicines by patients. So we can give the definition of good pharmacy practice according to the international documents. The good pharmacy practice, in other words, GPP, is the practice of pharmacy that responds to the needs of the people who use the pharmacist's services to provide optimal and evidence-based care. To support this practice, it's essential that there be an established national framework of quality standards and guidelines. Today, of course, today we say about international standards and guidelines, and of course, in our senior courses, we also will study another international guidelines. Yes, because international guidelines are universally for every country. Yes. And also we will consider the legislation of the Russian Federation about working of pharmacies in Russia. About requirements of good pharmacy practice, we can say that the good pharmacy practice requires that a pharmacist's first concern in all settings is the welfare of patients. Yes, it's very important. Also, the conception of the good pharmacy practice requires that the core of the pharmacy activity is to help patients make the best use of medicines. Yes. Fundamental functions include the supply of medication and other healthcare products of assured quality. For example, in our department, on the 4th course, we will study the working with medical devices. Yes, for example, thermometers, glucometers, tonometers, etc. Yes, because it's very popular goods for our patients in pharmacies, and we, as pharmacists, should know how these goods work. Yes. And also we can say that fundamental functions of good pharmacy practice includes the provision of appropriate information and advice to the patients. Yes, consulting of our patients in the conditions of public pharmacy, community pharmacy. Yes. Administration of medication, especially non-prescribing medicines. Yes, for example, because in every country, there are non-prescribing medicines which can be sold in the pharmacy by pharmacists without physician's prescription. Yes, so it's very interesting medicines, and we will study these medicines in our senior courses. And of course, fundamental functions of pharmacies and pharmacy in all include the monitoring of the effects, including side effects of medication use. Yes, yes, please come in. Also, we can say that GPP the good pharmacy practice requires that an integral part of the pharmacist's contribution is the promotion of rational and economic prescribing, as well as dispensing medicines to our patients. Also, we can say that the system, the conception of good pharmacy practice requires that the objective of each element of pharmacy service is relevant to the patient. Is clearly defined and is effectively communicated to all those involved. Multidisciplinary collaboration among healthcare professionals, including, of course, pharmacists, physicians, nurses, is the key factor for successfully improving patient safety. And let's speak about basic roles and functions and activities for pharmacists according to the international documents. Yes. So nowadays, the two large medical and pharmaceutical organizations, the International Pharmaceutical Federation, FIP, and the World Health Organization recommended that national pharmacy professional associations consider the following roles, functions, and activities for pharmacists where appropriate. Of course, this system there is in Russia too, of course, and we in Russia we also have the national system of regulation of working pharmacies and other medical and pharmaceutical organizations. Also, of course, we can say about basic roles of pharmacists according to the international documents by FIP and the World Health Organization. And of course, I can say that these roles are universally for every country, and these are the basic roles and functions for pharmacists as healthcare workers. Yes. So role number one: prepare, obtain, store, secure, distribute, administer, dispense, and dispose of medical products. And this, according to the international documents, this first role includes several functions. The function number one, of course, is prepare extemporaneous medicine preparations. Yes. Maybe someone from you was in our special building of pharmacy faculty during this educational summer school, and in this department, on the Department of Pharmaceutical Technology, teachers showed the basic principles of preparing medicines, yes, several days ago. Yes, so we can repeat that the one of the basic function of this role is prepare extemporaneous medicine preparations. And what means extemporaneous medicine preparations? Yes, extemporaneous medicine preparations are preparations which prepare in the conditions of pharmacy. Yes, so pharmacists in pharmacy, in community pharmacy, or in hospital pharmacy can prepare some medicine preparations by his or her hands. Yes, and you can see this process on the photo. Yes, the process of preparing extemporaneous medicine preparations. Yes, and of course On this photo, you can see the process of dividing medicines from one large package to a special little package for dispensing this medicine to patient. Yes. Yes, this process of dividing of medicines. The function number 2, obtain, store, and secure medicine preparations and medical products. Of course, every medicines should be stored in pharmacies in several conditions. Yes. Firstly, we as pharmacists, as future pharmacists, should consider, should take into account the temperature regime for storaging of medicines and another conditions for storaging of medicines. And of course, we will study these temperature regions. Yes, for example, the most of medicines, of course, can be stored in usually conditions. Yes, for example, yes, in the normal temperature, yes, about 20 or 25 degrees by Celsius. Yes, but some medicines, for example, immunobiological medicines, including vaccines, for example, should be stored in the low temperature, as usually immunobiological medicines are stored in the temperature or in the temperature at from 2 to 8 degrees by Celsius. Yes, of course, we will study these legal rules in our senior course too. And also in this photo, you can see the process of storing medicines in community or hospital pharmacy. Yes, on this photo, pharmacists put medicines in the basic places of storing medicines in the pharmacy. And of course, after preparing, after getting medicines, we as pharmacists should dispense of medical products to our patients. Yes, it's very interesting process. Yes, because during in this process we can consult of our patients about maybe about contraindication to using this medicine, for example, about storaging of this medicine in several conditions, and another aspects for good using, optimal using of medicines. Yes, and also, for example, we will study this course. Yes, we have in our pharmacy school, in our pharmacy faculty, we have the special course of pharmacy of pharmaceutical consulting and informing of our patients, and we will study this course on the third course, yes, with our students. And also we can say about the rule number 2 yes, because we are speaking about basic rules and functions for pharmacists. Yes, so let's speak about the rule number 2, provide effective medication therapy management. We can say that medication therapy management is a distinct service or group of services that optimize therapeutic outcomes for individual patients. Also, we can say that medication therapy management services are independent of, but can occur in conjunction with the provision of a medication product. And about functions of this rule. Firstly, we can say about assess patient health status and needs, because you can imagine the situation. Yes, for example, you are a pharmacist and you are working in pharmacy, and the patient come in your pharmacy. Yes, of course, in the process of consulting of this patient, we should understand his health status and needs. We can understand that this patient need to buy in our pharmacy, for example. So we can say that pharmacists should ensure that health management, disease prevention, and healthy lifestyle behavior, because we, according to the international rules and in Russian Federation too, by Russian legislation, pharmacists have the right to consult their patients about healthy lifestyle too. Yes, healthy lifestyle behavior. Because you understand that the healthy lifestyle behavior is very important for good health of our patients. Yes. So pharmacists should ensure that health management, disease prevention, and healthy lifestyle behavior are incorporated into the patient assessment and care process. Also, pharmacists should acknowledge unique patient considerations, such as his educational level, cultural beliefs, literacy, native language, and physical and mental capacity in all individual patient assessments. Yes, for realizing this function in our pharmacy school, pharmacy faculty, we have the special course of pharmaceutical bioethics. Yes, and we will study with our students the course of pharmaceutical bioethics in on the second year of education. Yes. Also, about next function, yes, manage patient medication therapy. It's available in situation. We can say that pharmacists should maintain access to an appropriate evidence base relating to the safe, rational, and cost-effective use of medicines, such as reference books on medicines, scientific journals, of course, national essential medicines lists, and standard treatment guidelines. Yes, because of course, every country has own special medicines lists, of course. Yes, so during of our education, we consider with our students firstly the international guidelines about medical therapy. For example, we consider with our students the essential medicines lists by the World Health Organization. Yes, the most important list of medicines. Yes, because the World Health Organization has prepared the several medicines lists, for example, for adults, for children. Yes, essential medicines lists for children, and the World Health Organization with other medical organization, for example, UNESCO, prepared the several lists of medicines and medical devices for using in the emergency settings. For example, in the situations with natural or technical disasters. Yes, it's very important direction of working medicine, it's very important direction of working physicians and pharmacists. Yes, and of course, we also studying with our students the main principles of organization of working pharmacists in the conditions of emergency situations, for example, in the natural and technical disasters. Yes. Also, if it's available for pharmacists, pharmacists can monitor patient progress and outcomes. For example, pharmacists should consider patient diagnosis and patient-specific needs when assessing patient response to medicine therapy and intervene if necessary. Of course, this function available in some countries. Yes, especially this function is more popular, we can say, yes, in countries when pharmacists mostly work in hospital pharmacies. Yes, because in the hospital pharmacies, pharmacists can use some ways. Pharmacists have some ways to diagnose to choose, yes, the patient condition and about using several kinds of analysis. Yes. But because mostly we say about the community, yes, the public pharmacies, yes, pharmacists also, of course, can monitor patient progress in public pharmacies too. But of course, you understand that in a public pharmacy, yes, we cannot use the special procedures for indication of patient health, yes, but we can consult our patients, we can ask our patients about their conditions before treatment and after treatment, and so taking into account this information, we can give information about patient progress in treatment. Yes. Another function is provide information about medicines and health-related issues, of course. We said about it, of course, we can say that pharmacists should ensure that in every pharmacy there is a suitable place for discussing confidential information with the customers and patients. Yes, also in the Russian Federation, we have the special order of Ministry of Health of the Russian Federation, and in this order of Ministry of Health of the Russian Federation consists the special legal rules, yes, about opportunity to organize in every public pharmacy the special places for discussing with our patients in confidential situation. Yes, because you understand that some patients can have the confidential information about their health status, and such patients usually want to talk to pharmacists in pharmacy in the confidential office, for example. Yes. Yes, and don't worry, please. Yes, we should consider another some roles and functions, and after this lecture we can show the video with work of public community pharmacy in USA, because yes, this video file will be in English. Yes, it will be more convenient for you, and we can say that nowadays, yes, we can say that the work of public community pharmacies in USA and in the Russian Federation are largely similar. Yes, because yes, you will watch this video. Yes, and about about role number 3, maintain and improve professional performance. Yes. According to the international rules, pharmacists should plan and implement continuing professional development strategies to improve current and future performance. Yes, in Russia too, of course. Pharmacists in the Russian Federation should improve their qualification and should use the conception of continuing professional development. Yes, so the concept of continuing professional development, CPD, can be defined as the responsibility of individual pharmacists for systematic maintenance, development, and broadening of knowledge, skills, attitudes to ensure continuing competence as a professional, healthcare professional throughout all their careers. Yes, all years of their careers. And also we can say that pharmacists should engage in preventive care activities and services. For example, pharmacists should engage in preventive care activities that promote public health and prevent disease, including in areas such as smoking cessation, infectious and sexually transmitted diseases. Yes, because in the community pharmacies, we also can consult our patients about this aspect, of course. Also, pharmacists should provide point-of-care testing where applicable and other health screening activities for patients at higher risk of disease, if it's available in such countries, of course. For example, in Russia, pharmacists can consult patients and can sell to patients their special medical devices for express testing. Yes, for example, there are express tests for indication of several infectious and non-infectious diseases by using of sample of blood, urine, saliva, and other biological liquids. Yes, it's available. It's very interesting development, and we, for example, we will study with others, with our students, such medical devices, express tests on our senior courses in our department. Yes, we have. In our pharmacy faculty, we have the special course of medical devices and medical and pharmaceutical merchandising. Yes, and in this course, we will use medical and other devices which can be sold in pharmacies by pharmacists. Yes, and last function number 3, comply with national professional obligations, guidelines, and legislations. Of course, pharmacists should take steps to ensure that they comply with the provisions of a national code of ethics for pharmacists. Yes, for example, international codes of ethics for pharmacists we also studying with our students on our second year of education in the course of pharmaceutical bioethics. Yes, it's very interesting too. So about conclusions of this lecture. And after this lecture, we will show the video file about the working of public pharmacy in USA in English. Yes. And about conclusions of this lecture, there are 4 main roles where pharmacists' involvement or supervision is expected by society and the individuals they serve. Firstly, prepare, obtain, store, secure, distribute, administer, dispense, and dispose of medical products. Secondly, provide effective medication therapy management to our patients. Thirdly, maintain and improve professional performance. And fourthly, contribute to improve effectiveness of the healthcare system and public health at all. Yes. In conclusions, yes, we can say that these roles may vary for each individual pharmacist in every country, depending on their practice responsibilities. And achieving specific standards of the good pharmacy practice for each nation, for each country. Within these guidelines may require considerable time and effort. And of course, we can say that pharmacists as healthcare professionals have a duty to begin the process without delay, of course. So let's start. We finished this lecture, and let's watch the video about the working of the public pharmacy in USA. And I will repeat that the working of public, especially the working of public pharmacies in the United States and in Russia are largely similar. Yes. For example, of course, we have for our students the practice in pharmacies, and in senior courses, yes, we go to our students in the pharmacies of our city, yes, of the city of Ufa. So let's watch this video file about the working of pharmacy in the USA.
Depending on what area of pharmacy you work in, there will be different expectations for the pharmacy technician. Here in community pharmacy, especially in an independent setting, we have huge expectations for the pharmacy technicians. They are, in essence, the pharmacist's right hand, and they help guide us through every step of the process of making sure somebody gets medication and that it's correct. In this independent setting, the technicians are responsible for not just filling medications and data entry, but they also have the opportunity to look at third-party reimbursements and correct pricing. They're also involved in inventory management and ordering, also ordering of chemicals and durable medical equipment, and especially things like Medicare billing and reimbursement by Medicare for durable medical equipment. In this independent community setting, the technicians have wonderful opportunities not only to advance themselves in many aspects of pharmacy practice, but the rewards that they get both from their customers and from the things that they do on a daily basis is wonderful.
This is where I receive all new prescriptions. We prioritize by the color of the baskets. White baskets are for people who are not necessarily waiting, coming back later, maybe things we're just putting on hold. But that's what these are. If I receive a red basket, that means somebody is waiting. So I always take a red basket first. In this basket will be important information like the prescription, obviously, any new insurance cards, photo identification for certain types of medication, and I'll go from there. The most important part of the prescription, the doctor's signature, and then the patient's date of birth. There are many patients with the same name, same middle initials, everything like that. So the easiest way to identify them is by their date of birth. So that's where I'll start off. So I've entered the patient's date of birth, which has pulled up their name and their entire profile. Here you'll see all the drugs they've gotten in the past, any list of allergies, things like that. So I received a new prescription. I do see there's new allergies on there, so I can double check to make sure through in their patient profile that this medication doesn't interact adversely to what they have. Then I'll go back and I'll just start entering the prescription. I'll enter the doctor's name and then the drug. The drug will give me a list of many generics and things like that, but I can go ahead and type that in, type the quantity, type the directions, make sure I'm choosing the correct NDC. A lot of times there are NDC, many generics for one brand. Some patients prefer certain NDCs, so I'm going to double check to make sure this is the only one we have for this drug, so I'm going to choose that one. I'm going through making sure that the directions are the same, the day supply is calculated correctly, that it's a written prescription versus an e-scripted prescription versus a phone-in prescription. That's all very important information. The day the prescription was written will also be very important as far as expiration of the prescription if there are refills. And then I go ahead and make sure if they have insurance, I want to make sure I'm processing it through the correct insurance. They don't, so we set a cash price for them. So once I processed all their prescriptions, I go ahead and print a label. The label will print up here. I'm going to put the printed label with the prescription, and then I'm going to pull the drug to give to the pharmacist to count out and to verify that my typing is correct, that the doctor is correct in medication, and then he dispenses from there. All right, so I've found the correct drug. On the label is the correct NDC, which matches up with the bottle. I'm going to make sure that is the correct NDC, and then I found it. So after that, I just give it to the pharmacist. And from then the pharmacist will double check my counting, count up the prescription, and it'll be ready for the patient to receive. When receiving a C2 prescription, there are a little more checks and balances. First, we're going to need proper photo identification. That is a valid photo ID, could be a driver's license, anything with a passport, anything with a picture ID of the patient that is current, up to date. The prescription next is also very important. The doctor's signature, the doctor's DEA number, and the date, the way they've written it, everything has to be written out very specifically. So once we've verified the doctor's DEA, then we make sure they're within date range. If they are within proper date range to fill the prescription, we go ahead and fill it. The pharmacist then will take it over. They will use the E4 system to check to make sure all controlled substances are within proper dates of filling them. We do make a photocopy of the identification to attach to the prescription to be filed away for each C2, along with the E4 report.
Pharmacy technicians are responsible for properly triaging insurance. And there's different types of insurances. There's commercial insurance, there's Medicare plans, and then there's state plans like Medicaid. There's some significant differences amongst those plans. It's expected that the technician know how to properly bill those and how to address the problems that may arise when they're trying to bill those insurance companies. It's not just problems with the insurance themselves, but with the insurance company requiring prior authorization or not covering certain products. So part of our expectations here are that the pharmacy technicians know how to bill those properly. We do a lot more Medicare billing here and require more out of our technicians as far as their knowledge with third-party plans. In community pharmacy, some of the technicians' duties may include compounding. In this particular location, we do allow our pharmacy technicians to compound certain creams, capsules, suppositories, and other things that they may need to do. The majority of this is stock supplies. This will vary depending on what pharmacy you're in and what they allow their technicians to do. The technician is responsible for doing the math calculations, for actually putting the compound together, and it's double checked by the pharmacist before it's put together and again after it's put together.
Today we're going to make Proxicam capsules, and first we're going to start off with all our supplies. So I'm going to grab some gloves. Okay, we're going to put on the mask. Okay, now we pulled the drug. So we're going to write down what we're making today. We're doing Proxicam 10 milligrams. So we're going to write down the drug, date, compounding, the ingredients you're going to use, the lot number, expiration, manufacturer, and the lot they want to fill in. Okay, all right, we're going to go ahead and do the calculation. So we're doing 10 milligram capsules, okay? And we're going to times that by 100 because that's how many we're going to do in quantity. So that comes to 1,000, and then there's 1,000 milligrams in a gram. So that's how you get your gram. Milligrams, caps, and this will be 1 gram. This is milligrams with a total, and then we go out to 1 gram. These are 10 milligram capsules. We're doing quantity of 100, which equals 1,000, and you take 1,000 milligrams into a gram is 1 gram. We're going to weigh this out.
1,000 milligrams is 1 gram. the process of measuring
Close the sides because sometimes there's air when people walk back and forth.
You can see the electronic scale for measuring.
Very touchy. There you go. Okay, I'm going to pull the rest of the drugs that we're going to use, which is lactose. Okay, so these are all my drugs. Next, we're going to set up the capsule machine itself. So we're going to use, these are #1 capsules. We're going to load up the
After
machine.
preparing, it's need to package the prepared medicines to get this situation.
This releases the capsule. And it does a set of 50 at each time. You just want to push these down. Put this back on. And push it forward so you get the other 50 in there. I'll close up as I go. Make sure we don't spill anything. Okay, this comes off. Some of them turn upside down. Gotta turn right way. Cover on. You want to tighten the top. And seal this up. Okay, I'm going to put the capsules back down here. Okay, we're going to take this apart. We're going to release this. I'll drop the capsules down.
Right. A packing stat is how many milligrams of drug fits into a certain size of a capsule. We're using size 1 capsules right now. Our filler is lactose. Our drug is piroxicam. So a size 1 capsule holds 315 milligrams of piroxicam. It holds 365 milligrams of lactose. We're only filling our capsule with 10 milligrams of drug, which is the piroxicam. So therefore, that's 3% of that capsule will be full. So 97% of the capsule will contain lactose. 97% times the 365 milligrams that capsule holds times 100 will give us the amount of lactose that we need to add to the batch of 100. The technician will weigh out the amount of lactose that she needs and the amount of drug that she needs and mix it in a mortar and pestle.
Okay, now we're going to mix the piroxicam with the lactose.
Lactose is the additional component of this medicine. You can see the process of measurement.
Oh, yeah. I'm going to go call a pharmacist. Jessica, you come check my work, please.
I checked the technician's work to make sure the calculations are correct and that she's using the proper equipment and has everything set up. I'll do another check after she's completed this. Usually the technician and I will have a discussion during the time that I'm checking to go over the process of compounding the medication. Okay. You're making 100 capsules, so add some of the lactose, then your drug, and the remaining part of the lactose, mix it until it's uniform, and then you can make your capsules. And then just call me when you're done.
Okay. All right. Okay, we're going to put a little bit of the lactose in. The drug. And mix this around. Make sure the drug gets in there. I'm going to add the rest of the lactose. Try to get equal parts. And the rest of the lactose. Okay. Next, we're going to put them into the capsule machine. I'm just going to dump that here.
And the process of packaging this medicine in different countries.
We'll start into the capsules. And we just give it a little tap. You take this little tool just to push down the drug so we can get the remainder inside the capsule. Okay. Okay. Put the lid back on. Loosen this back up. Taking it, squeeze the plates together at the top. And we're going to seal them. This pushes the capsules together. I have one capsule that didn't pull out when I put them together. So what I'm going to do is I'm going to lightly pull up on the bottom. I've got the total ingredient. And I'm going to cap it, push it right on there. So this way we didn't lose the capsule itself. We're going to open this up. Knock them out. That's the finished product. So then I will get the pharmacist. Jessica?
Part of the checking that I do is to make sure that each dosage is uniform. So I'll check to make sure that she has the same amount of powder in each capsule. In this particular case, the technician's technique and process that they use is very important in making sure that the final product is uniform. In this case, when you saw her adding the drug and mixing the drug together with the inert substance, which was the lactose, she used a process called geometric dilution. And that's where she added some of the lactose first, approximately 1 gram, the same amount of the drug. She mixed that together and then added the remaining part of the lactose. And that helped her get a very uniform distribution of the drug in the entire dilution. Also, the process that she used in putting it into the capsules, she used something called a punch, which punches the drug and the lactose into the capsule. And she does that because it helps her get an even amount of drug in every capsule.
And
There's the finished product.
prepare capsules.
When inputting nursing home prescriptions, they're the same as typing on a regular prescription, except that you need times and what the medication is for. And how they're taking it, such as orally, via G-tube, and once or twice a day. So in this instance, I'm filling a prescription for potassium, and it's by mouth twice a day as needed for a supplement. When filling nursing home prescriptions, they're in these little cards with specific numbers per dates. We put one in each spot, so when they get to the nursing facility, they can just pop it out. And since there's 30 days in September, right? Okay. So we will only fill up to the 30. And then our labels go on.
In USA, this process is more spread than dividing
Set it aside.
large bottle to a little package for dispensing.
After the process of packaging and the pharmacist checking our work, we keep these separate from the regular customers. Nursing home is different, where we deliver sometimes, or the homes come and pick them up. So they're separated.
I'd like to explain how we do our ordering for our stock up front. It's the same in the back. When we sell an item, each of our items has a sticker from our supplier. We take that sticker off and put it on a piece of paper so that when we get a chance and we don't have a customer in front of us, we'll go back and order from the order computer. Not every store does that. Some stores do an electronic where they will come out and scan their shelves. They might do that every evening or possibly whenever they have a free moment. Then that gets automatically sent to the supplier. When we get a chance, we take that information that we have put down, and the prescription area also does the same thing. They take empty bottles also and put them in a container so that we can scan them. We do this periodically through the day so that the pharmacist has a clear workspace. And in our instance, the orders are sent at specific time. It's just automatic.
The orders received by the technician, we receive the actual product in the store. That product is checked against the invoice. And that's done for accuracy in case something's been shorted or in case something has been billed to us incorrectly. The product is stickered with the cost of the medication and the date that it was received, as well as a reference to the invoice number. And then those products are placed on the shelf. There are a couple of exceptions. One is controlled substances. Class 2 controlled substances are handled a little bit differently. They're always separated when they come in from the wholesale house from the other inventory. That's what this is in front of me. There's a form called a 222 form. It's a form that goes to the DEA, and the technician is responsible for checking the invoice against our 222 form, which is our ordering form. They put the number of packages received, they sign it, and a copy of this goes to the DEA. Depending on the pharmacy that you're working in, receiving inventory may be done a little differently. The class 2 medications, however, that form is standard and that won't change between pharmacies. Sometimes we get refrigerated or items that have special storage. And those are shipped differently, and those are always a priority when they come into the pharmacy so that we don't store them incorrectly. The technicians in this setting are responsible for pulling expired medications off the shelf. We pull 3 months ahead of time, and then there's a company that will give us credit for those returns and destroy them. For instance, this medication is going to expire within the next month. So our first priority is to get it off the shelf and away from our inventory that has current dating. We separate these by putting them in a bin, and this gets located away from the regular inventory to be sent back and destroyed.
Hi, can I help you?
Yeah, I was looking at my mom's prescription and stuff, and I noticed that there's a couple things wrong on here. You have the doctor's wrong, and I'm not happy. I mean, how do I know that everything is correct? I mean, the medicine itself, and I mean, I don't know how many times this has happened before.
Let me check the prescription for you while you have a moment.
Okay.
Okay. Okay, I've got the prescription here for you. Evidently, the physician's assistant was in and signed the order for you, so that's the physician's assistant for the doctor. So, and I double checked, the quantity is correct, also the strength of the medication is correct. Did you have any other concerns?
No, I just want, they gotta be careful on double checking and stuff that they have. And I mean, I want to make sure that this is my mom. I want to make sure she's got the right medicine. And,
I don't blame you. If there's any question, don't hesitate to call.
She's old. She doesn't know what to look for and certain things. And I looked at hers and I was like, that's got the wrong information on there.
Sure.
I want to make sure that, the right medicine and stuff is in there.
If you'll just let her know that the physician's assistant is the one that signed, the physician's assistant works for the same doctor that she sees. So, and as I say, if she has any questions or if you have any questions, please don't hesitate to come in or call us and we'll be happy to review it for you.
All right, thank you.
There you go. Good morning.
Hi.
Have you been with us before?
No, first time.
Okay. I need to get some information from you. I need to get an address.
8191 Marguerite Circle.
I need a date of birth.
9/26/50.
Any allergies to medications?
Penicillin.
You said you had an allergy or a reaction to erythromycin.
My stomach.
Oh, right. Would you like childproof or non-childproof, Liz? Easier then. And would you care to wait for this, or would you like to come back? All right. It'll be probably about 15 minutes or so. You'd like to have a seat or be right back. You were asking about free of dyes and fragrances. We do have a line that a lot of the dermatologists are looking at nowadays. We have this particular item. I don't know whether you're looking for a cleansing bar, but they also have cleansing bars that are free of dyes and fragrance.
It'd be good for my wife, but how about a smaller size just for me today?
We do have a smaller size. It comes in a tube. It also comes in a pump.
And it's dye-free and fragrance-free?
It is.
Oh, great. That would be good for her.
Okay. You're all
But
set.
she's
And
also
you-
got some bladder irritation. She wanted me to ask you if you had anything to help her urinary discomfort.
We do have things for the urinary tract infection, but these are items that I would like to have a pharmacist come out and check with you on, because he might have specific questions as to how this is going to affect the patient. So if you have a moment, I'll see if I can get a pharmacist for a consultation.
That would be great. Thank you so much. I appreciate that.
I've picked up your father's information. It looks like we're looking today. Does he need both the wafers and the pouches? Okay. And I do have the numbers that he has been getting in the past. So let me see what we're picking out here. All right, and I do need to double check, has any information changed for your father? Has his address changed or phone number? What address does Medicare have on record for your father?
I think
it should be the same, okay. Then, and the doctor is the same as the last time, no changes have been made there.
Do I have to give me his ID card today?
We have his Medicare number, you gave it to us originally when we signed it up, so we are all set with that. And we do have a refill for now. We will need to get a refill from the doctor the next time that he comes in. So if he's going to be seeing the doctor, if you could bring that in with us, with you when you come. And I will have you, I'll fill out the paperwork, have you sign it. We do need to run it through the system so that it can be sent on to Medicare so that any reimbursement will come to you.
Oh, thank you so much.
We've got your prescriptions ready for you. I've gone ahead and scanned you out, but I need to get from you, since it's the first time you've been with us, for the HIPAA information, I need your signature. Okay, and what I need you to do is sign right here for the HIPAA information. And after that, if you'll hit done, and then I'll also need your signature that you've picked up your prescriptions. Okay, you're all set. I have HIPAA policy, and I'll give you that since it is the first time you've been with us. If you have any questions at all, this is that we do not sell your signature or anything to you. You're more than welcome to look at it. And if you have any questions, please don't hesitate to give us a call. Do you have any questions for the pharmacist? All right. If you have any questions at all, please give us a call and hope you have a good day.
Okay, very good. And so you watched the basis of the work of public community pharmacy. Yes, so of course, with our pharmacy students, we on the senior courses, we will study the specialized courses such as pharmaceutical consulting and informing of patients, pharmacy administration, pharmaceutical marketing, pharmaceutical management, and other disciplines. And of course, we are going with our students to pharmacies of our city, Ufa, to practice. Yes. And also today with us our resident, Bishoy Khalil. Yes, our resident of our department. Yes, and if you have any questions, you can ask me or our resident Bishoy. Yes. Okay. Yes. Our lesson is over. Yes. Thank you so much for coming and joining to our lesson. I sincerely wish you a good health, good success, good luck in everything. Yes, and see you in our future lectures and lessons. Yes, good luck. Goodbye.
Thank you so much. Goodbye.
Thank you so much.
Goodbye.